Dr. Terasaki had a vision for improving the lives of transplant patients worldwide. At TIC, we're continuing his quest for discoveries and innovations that will lead to a better understanding of the mechanism of allograft rejection. Through international partnerships, it is our goal to develop new pathways and tools enabling transplant centers to better manage their patients. Visit our website at www.tictx.org to learn more.
Welcome to the Ashi Virtual Global Annual Meeting Scientific Awards presentation. I'm Medhat Askar, and I'll be your moderator. Our first scientific award is the Paul Trasaki Clinical Science Award. The Paul I. Trasaki Clinical Science Award was established in 2003 to honor individual, group, or institution in recognition of significant accomplishment and/or contributions to the fields of clinical transplantation, histocompatibility, and immunogenetics. The 2020 Paul Trasaki Clinical Science Award goes to Dr. Alexandre Loupy. Alexandre Loupy is a professor of nephrology and epidemiology, kidney transplant departments at Necker Hospital in Paris. His research focuses on new methodological approaches of multi-organ transplantation. It covers allograft allocation, rejection, populational sciences, and risk stratification. Professor Loupy defended two, not just one, PhD degrees in cell biology in 2011 and biostatistics in 2014. Since 2015, he is heading the Paris Expertise Center for Organ Transplantation. Professor Loupy is an expert for the United States FDA. He is an active member of the American Society of Transplantation, an associate editor of the American Journal of Transplantation, and the recipient of the Clinical Science Investigator Award from the American Society of Transplantation in 2017. He was awarded the National Academy of Medicine Award for his work on renal transplantation, anti-HLA, graft rejection, and the biomarker identification. Professor Loupy is also heavily involved in the French Society of Transplantation and in the European Society of Transplantation. In the past years, he has obtained several grants to support implementing cutting-edge diagnostic tests at large scale. Professor Loupy is the principal investigator and coordinator of the International H2020 EU Train and the National RHU KTD Innov projects. I can go on and on and on, but without further ado, please welcome Professor Alexandre Loupy.
Dear Ashi Awards Committee, it's a great honor for me to receive the 2020 Paul Terasaki Clinical Science Award. I am very excited to receive it. Winning this award has a particular test for me and meaning, because I'm, first of all, a big fan of Paul Terasaki's accomplishment for patients and also his personal journey. And in fact, I have a picture of him on the wall right next to my desk. And I also wanted to extend my most sincere thanks to the Ashi for their exceptional work. How I think of the society and its importance for the transplant field. So this award truly gives me great encouragement to maintain my enthusiasm in the future and also to continue to work with great people like you, gain knowledge, and last but not least, improve myself. Thank you again for this award, and it's a big honor for me to receive it. Hello everyone, and hello from Paris. I'm very honored to give this presentation regarding the Paul Terasaki Award. And my talk will be covering some aspects of anti-HLA antibody integration into the transplant universe. We'll talk about current challenges, the journey, and the unmet needs for proper approaches. So the story started in my hospital, Necker Hospital, back in December 1952, and you see that this ambulance is carrying the young Marius Renard, He was a carpenter and fell from a stair, and he was sentenced to death because he had a unique kidney, and this kidney had a severe hemorrhage. So the attending physician had to remove this kidney. And Mario Sonar was transplanted, and he had the transplant from his mother. Unfortunately, it was the first transplant, and no one knew anything about histocompatibility at that time. And Mario Sonar died after 2 weeks post-transplant. And what you can see here is the first biopsy of human acute rejection from Mario Sonar in January 1953. And obviously, massive rejection going on. So these are the early years in Necker Hospital in Paris, and you've all heard about Jean Burger. And Jean Burger in 1954 started to think about the fact that we should stop the transplant programs. There were too many deaths, too many failures, and the program was in danger. And you can see here the Carré Necker, a picture taken in 1953, and those 3 windows actually where the rooms where transplants were performed, and the first kidney transplant patients were staying. And this is a quote from Jean Burger that I like a lot, because he found that complement was normal and the gamma globulin fraction was elevated. It is therefore possible to hypothesize that this increase was due to antibodies against the transplanted cell. So maybe that was one of the first mention of DSA by Jean Burger. So things have changed since then. And obviously the head of the department is not the same one. And Christophe Lejean now is trying to transplant everyone, and we are crossing the HLA barrier. We're doing high-risk kidney transplants. And interestingly, you can see that in 2020, the Carré Necker still looks exactly the same as in 1953. Not sure that we have to be proud of it, but anyway, some things change, others don't change. But obviously, what happened in Paris in the early years was Jean Dosset and Paul Terasaki. And Paul Terasaki, as you probably know, and it's mentioned in Jean Dosset biography, spent some time in Paris, and they worked together and made a very important connection. So let's say that Paris is perfectly a perfect match for antibodies, and you see here the perfect combination and the alignment between the alpha tower and an antibody. So this is about now my transplant journey, how it started. And actually, it started in Réunion Islands, the place where I was born and raised. That was me in 2003, and where I am now. And I'm still, enjoying to spend holidays in Mauritius or Réunion Island, and I read books. And this is the Jean Dosset Nobel Prize of Medicine, Clin d'œil à la vie, this book, which is this big HLA adventure. And this is really a nice book. Unfortunately, it's only written in French, but believe me, that's a very cool book. So my goal when I was a resident and when I started to work in a transplant department was maybe to think about achieving a more precision medicine assessment in solid organ transplant, meaning measuring objectively relevant pathophysiology components to revealing mechanism, risk stratification, best choice for therapy, response to treatment, and surrogate marker for outcome. But the thing is that we started to understand that DSA was kind of chaos. But instead of being disappointing, we tried to organize the chaos. And of course, when we talk about DSA, we talk about pathogenicity, distant clinical expression, distant phenotypes, distant timelines, distant kinetics, distant disease process, and the problem with non-HLA antibodies and complex phenotypes, and last but not least, response to treatment. So clearly the global approach was to try to connect the dots and try to connect people around the same goal, better understand the deleterious power of anti-HLA antibodies. So we started to connect classical histopathology with immunochemistry and presence of circulating anti-HLA DSA. And of course, the idea was that integrative approach was really mandatory. And you can see here in this slide the landscape of HLA biomarkers, chemistry, clinical history, light macroscopy, immunohistochemistry, immunofluorescence, electron microscopy, and molecular diagnostics to better address diagnostic activity, staging, stratification, and treatment. So this is my journey. When I was a resident, we studied and we worked hard and trying to better identify antibody-mediated rejection. And we published this paper back in 2009, the first identification of subclinical antibody-mediated rejection. So this was a very high-risk cohort of kidney transplant recipients. And we identified patients which were perfectly stable at 3 months and 1 year post-transplant, which were transplanted across and with presenting a circulating anti-HLA DSA at the time of transplant. And what we could see is that those patients, they had evidence for antibody injury, meaning microcirculation inflammation, presence of C4D deposition in capillaries, and evidence for serologic antibody. They were DSA positive. So we made it into the cover of AJT, and I was a resident. I was super proud of this paper. I think it was cited more than 400 times. And it was really an important piece in my career to step into the better identification of the spectrum of antibody-mediated rejection. So we followed and we identified those muted form of antibody-mediated rejection, and we identified what we call C4D negative antibody-mediated rejection. Using the same high-risk cohorts implemented by new patients and a huge and stereotyped protocol, immunosuppressive drug protocol, we identified the fact that C4D status in patients was fluctuating with time. There was wide swings in the C4D deposition in capillaries when we took the biopsy for cause, the protocol biopsy at 3 months post-transplant, and the protocol biopsies at 1 year post-transplant, and the biopsies performed at later time points. And interestingly, we found that despite the presence of microcirculation inflammation and circulating anti-HLA DSA, more than 50% of those biopsies did not present any C4D deposition in capillaries. So in other words, it was the identification of a muted and form of antibody rejection. So we moved from 2011 to 2013 and 2016. And those 3 years really paved the way towards the extension of the spectrum of antibody-mediated rejection. We identified, remember, in 2009, subclinical antibody-mediated rejection, and we moved into the epidemiological demonstration and characterization of this entity in terms of association with outcome, independency with different other parameters, and renal function, proteinuria, time post-transplant, and scarring. And you have here the summary of the study design. But we also identified in this paper the fact that not only subclinical ABMR, but the presence of acute T-cell mediated rejection was associated with the later development of de novo DSA. This association from TCMR to the development of de novo DSA was described in this paper and further demonstrated by many other teams. And again, this paper came with a very nice editorial written by Peter Nickerson and David Rush. And I really like the title, Begin at the Beginning to Prevent the End. I think it was important because it was the notion that ABMR was not an episode. ABMR was a process. And we had to think about the way we could identify this process as very early stages to be amenable to therapeutic interventions. Another important component for the refinement and the extension of the spectrum of antibody-mediated rejection was the fact that vasculitis was primarily considered to be a T-cell mediated rejection component. And in this paper, we took a large cohort of really well-characterized transplant patients, kidney transplant patients, with rejection. And we found, using unsupervised analysis, the association between the presence of circulating DSA and the association with vasculitis lesions and the later development of chronic rejection and transplant glomerulopathy. interestingly, that was the first paper where we used those unsupervised approach to get rid of the preconceived ideas of the classification, because more than 40% of patients with vasculitis lesions were misclassified as being T-cell mediated rejection, and they actually had antibody mediated rejection. And the downstream consequences is that most of those patients did receive T-cell targeting therapies and not antibody-directed therapies, and maybe the consequences of their bad outcome. So this paper was released in Lancet with another editorial, and it really made the point of the extension that vasculitis was not the unique province of T-cell mediated rejection, and the extension of the spectrum of AMR was starting from a microcirculation disease to the harm of the macro vessels, and in this case it was vasculitis. We went a bit further and we asked ourselves the question whether there could be an acceleration of arteriosclerosis post-transplant related to the presence of circulating anti-HLA DSA. And I was glad to work with Dominique Nochely and Gary Hill in this paper published in JSN in 2011, which actually made the real clear point of a phenotype of dramatic acceleration of arteriosclerosis lesions with fibro-intimal thickening of the arteries post-transplant. And it was independent of the donor age. And we could draw this line, which is the normal progression of arteriosclerosis with age. And you can see that nice figure, which summarizes that. And we identified that in patients with post-transplant antibody-mediated rejection, there was a 28 years aging in the first year post-transplant of the fibro-intimal thickening of the arteries, which we call DSA-mediated acceleration of arteriosclerosis. So we had the Lancet paper with acute vasculitis and the continuum towards chronic vascular lesions, DSA-mediated acceleration of arteriosclerosis. We went a bit further and published in Circulation Research further on a much larger cohort where our hypothesis was that alloimmune response could actually induce local allograft premature arteriosclerosis independently of classical determinants of atherosclerosis and arteriosclerosis. So the study design is shown here. We had more than 1,000 patients, carefully phenotyped, assessment of donor cardiovascular profile, recipient cardiovascular profile, baseline transplant profile. We had the immunological profile, graft phenotype, and post-transplant profile with BMA, blood pressure, new onset diabetes after transplant, LDL value, triglyceride, antiagglutinin, the treatment, statin, CNI, and steroids, and many other parameters. And we could build a very nice model which could actually identify the part that DSA played in the acceleration of arteriosclerosis post-transplant after adjustment on those many parameters that can contribute to arteriosclerosis. So we found that there was a very important strength of this association. We found also the connection with distinct phenotypes that are summarized here. And this paper came with a very nice editorial from John Brownberg, and I like the title a lot, Alloantibodies and Allograft Arteriosclerosis, Accelerated Adversity Ahead. And there is a question mark here. So epidemiological demonstration of accelerated arteriosclerosis. And of course, you know, starting in 2011 and 2015 and 2016, we tried to ask ourselves whether this concept of acceleration of arteriosclerosis and increased vascular aging related to the presence of circulating DSA, that concept could be transported from kidney to heart transplant recipients. And these are two important papers we published with Jean-Paul and Patrick Gruval and the rest of the team, very talented and outstanding pathologists. And you see here these beautiful pictures, and also the cover that we made in NJT. And this paper, we think, were really important because they define the continuum between the presence of antibodies and the very late heart transplant rejection. And also we carefully phenotyped late failing heart allografts, and we found that there was an association between chronic allograft vasculopathy with antibody-mediated rejection in those late failing heart allografts. We are also glad to see that actually this paper made it in the top 10 papers released in 2016, and this editorial by Bruce Cavan. We are really pleased to find that those concepts in kidney could be seen in the heart. It was very important for us because it was a step into the extension of our approach in another organ. And you will see that now we are tackling some of the questions in lung and maybe liver, which is more difficult, but we are really happy to see that. So it became apparent that not all DSA were created equal, and beyond the simple presence of anti-HLA DSA and the specificity and also the characteristic and the properties of DSA could be important. So what we tried to do, we tried to build this important prospective and multicentric cohort where patients were carefully phenotyped, and we assess at the time of transplant and in the first year post-transplant at 1 year off, in the case of every single clinical event in the first year post-transplant, the properties of the DSA and whether the presence of C1q binding antibodies could be associated with a distinct phenotype, a distinct long-term allograft outcome, and how those movements and change between the status of the DSA could be associated with worse prognosis. We build a very strong epidemiological model to try to isolate the independent value of the presence of complement binding DSA for prediction of long-term allograft loss. It's a beautiful collaboration that we were lucky to have with Adriana Zevi in Pittsburgh in the US. And as you can see, the take-home message from that is that actually we found that independently of other parameters, the presence of complement binding DSA was associated with a more dramatic decrease of allograft survival, but also that non-complement binding DSA, as you can see here in the blue curve, were also showed an impaired prognosis as compared to patients without any circulating anti-HLA DSA. So we found that there was a gradation of risk and different phenotypes in those populations. So we wanted to go deeper into the detail and try to better connect the dots. It's always connecting the dots that we had in mind. And connecting anti-HLA DSA properties, complement binding activity, using integrative approach. And in this paper we published in JSN, we found that there was a mixture of IgG subclass, and we found that the IgG subclasses, the injury pattern and driving forces were more that IgG3, actually a combination of IgG1 and 3, were the driving force towards acute full-blown antibody-mediated rejection, while the presence of IgG4 was mainly driving indolent phenotype. So there was another dimension of understanding, despite the fact that it's very complex to detect and those subclasses. And again, it was a beautiful collaboration with Carmen and also Adriana Zevi from the team of Pittsburgh. A very important part of what we found was to address the question whether those properties and the monitoring of DSA properties from the time of transplant to post-transplant could improve the discrimination or the reclassification or the prediction of long-term kidney allograft survival. So we built another cohort, again, carefully phenotyped, where we assessed at the time of transplant, the model, which is the reference model, combining donor characteristics, recipient characteristics, transplant characteristics, immunological anti-HLA DSA characteristic, with different models, model 2, 3, 4, which incrementally assess and address the DSA detection post-transplant, adding to that their complement binding capacity and IgG subclass. And we found that actually when you monitor DSA post-transplant, you increase your reclassification of risk in terms of long-term allograft survival. In other words, there was a clinical value of monitoring this DSA post-transplant, and especially the beta characterization was associated with an improvement in the risk stratification of patients for long-term allograft loss. We could not address in this paper the proper kinetics for monitoring this DSA and the characteristic, but at least it was a demonstration that a very precise monitoring of DSA properties post-transplant was associated with an increment in the prediction and the performance of your model to predict long-term allograft failure. And then we started to understand, and we started this collaboration with Phil Halloran, and our goal was to better characterize those early onset antibody-mediated kidney allograft rejection related to patients who were transplanted across the HLA barrier. So they basically all had pre-existing and day zero anti-HLA DSA present at the time of transplant. And the hypothesis was that the molecular microscope system could add another dimension, not only to dissect the biological operational process that the eyes of the pathologist could not see, but on top of that could provide an additional feature for better re-stratifying patients. And in this paper, which we published in JSN, we could demonstrate that the addition of the molecular microscope strategy could improve the re-stratification in early antibody-mediated kidney allograft rejection. So again, it was a small cohort. That was the first step, but it was a very important argument towards the potential need of those complementary approach on top of the classical histopathology. And we also further refined the phenotypes, and we are now considering immunohistomolecular phenotypes and published this paper in JSN in 2017. And we found that there was not only distinct allograft phenotypes, molecular phenotypes, but outcome in regarding the fact that patients had pre-existing DSA-related antibody-mediated rejection or antibody-mediated rejection related to de novo DSA. So it was very important to tackle this phenomenon and dissect the deleterious impact that those antibodies could have depending on the fact that they were pre-existing or de novo. And again, as I say that we moved into immunohistomolecular rejection phenotypes, we also found that complement activating anti-HLA antibodies could provide another dimension for differential gene expression profiling. And you could see here that we had very top transcripts and top genes associated with the fact that patients had complement binding DSA or non-complement binding DSA. We found that the top genes model outperformed the standard histology model, so could add another dimension into precision diagnostics. And we also found that those complement ABMR selected transcripts could unravel different biological processes related to gamma interferon or INK related genes, macrophages genes, and also endothelial genes. Importantly, in that paper, we assessed in the eculizumab trial the distant association with the outcome, and the outcome was rejection rate regarding standard of care and rejection rate in complement inhibitors arm. And we found that actually the overall transplant population in the clinical trial were given complement inhibitors, and in a substantial fraction of these patients, they had non-complement binding antibodies. So the absence of response was not due to the inefficiency of the drug. It was due to a defect in the study design and in the metrics that were used to assess the patient outcome. So now we moved into 2017, and again, we published in Circulation this paper, which showed the gene expression profiling for the identification and classification of antibody-mediated heart rejection. And obviously, what you can see here is that we had histological phenotypes, DSA phenotypes, and molecular phenotypes. And this paper came out with a very nice editorial written by John Kobayashi. So we were able to find that the molecular landscape of antibody-mediated rejection in kidney could be translated in heart with a lot of similitude, but also some discrepancies. And actually that the current ASHLT-based classification for heart antibody-mediated rejection could be challenged by this approach and unravel distant level of disease activity and a potential for improving the ASHLT classification. So 2017, integrate gene expression crossing the diaphragm. What about 2018? We saw that it was finally time for synthesis, not only us published stuff around AMR, it was kind of a big deal at that time. So we decided to perform a very step-by-step and well-methodological executed systematic review and meta-analysis regarding DSA properties and risk of rejection and allograft failure. So we published in PLOS Medicine this meta-analysis combining everything that had been published around the subject without any preconceived ideas and taking into account every single study metrics regarding protocol and methodology, very careful sub-analysis and meta-regression in different subgroups, and also a very careful analytics on the respective association and a deep dive into all these papers that were selected based on a protocolized systematic review. And as you can see here, we found and we confirmed that actually overall, when we pull all the studies together and we assess also the heterogeneity between the studies, that complement activating DSA actually carried a poor allograft survival. It was true regardless of the test used, C1q, IgG3, C3d, C4d. It was confirmed in all organs, kidney, heart, but also lung, and interestingly, liver. It was confirmed regarding timelines, if the fact that the DSA was pre-existing, persisting, or de novo DSA. It was confirmed according to the different teams which have studied the subject. And last but not least, look at the association overall with the outcome, which is rejection and allograft failure, is 3.09. So there was a very strong correlation in this meta-analysis between the presence of complement activating antibodies and the risk of rejection and long-term allograft loss. So, time for synthesis, HLA-DSA, a ready-to-use biomarker for precision medicine. At least there was the accumulating evidence in the past 10 years that characterization and improvement in the characterization of antibody lead to improved risk stratification, empowerment of clinical trials, precision diagnosis, and addressing disease heterogeneity using complementary approach such as molecular assessment in the allograft, and last but not least, therapy efficacy. And this is also a synthesis that we published in the New England in 2018. And you can see here this huge collaboration between immunologists, nephrologists, heart transplant surgeons, pathologists, and all our network. And I'm very grateful that we could combine that together and build that aspect of antibody-mediated rejection across solid organs in kidney, heart, and lung, adding all the data and accumulation of knowledge drawn from population-based studies, addition of molecular approaches to better tackle the pathophysiology, and also the way that we could envision the landscape of the potential therapeutic targets and how we can improve the way that we are using these drugs to empower clinical trials, taking into account the different archetypes of antibody-mediated rejection and having a more pathogenesis-based approach of therapeutic interventions in antibody-mediated rejection. So, time for synthesis. This is how the BANF process actually could evolve, could take the lessons from the available converging results and data and extension of the spectrum of antibody-mediated rejection, better understanding the spectrum between acute, chronic lesions, active lesions, V lesions, C4d negative AMR, the fact that antibody was a process, the impact of DSA, but also other antigens, and also the importance beyond antibody-mediated rejection and the fact that T-cell mediated and antibody-mediated rejection could react as unison. And we are now better understanding the pathways between these entities and how to better characterize and treat our patients. Of course, all the molecular technologies and gene expression assessment via allograft help to unravel those entities. And I think that it's very important for the constant improvement of the BANF classification. And diagnostics in BANF is an evolving process from 2013, 15, 17. And I would like to spend a little bit more time on what happened in 2019, where the BANF Ashi. It was a very nice and entertaining conference with a lot of discussions around antibody-mediated rejection, of course. And I'm very grateful, and I would like to take the time now to thank all the very important people who have been instrumental in this conference and also in my career. And of course, you can see here the usual suspects, and Adriana, Michael, Marcus, Bob Corbin, Kim Soles, Jack Demetris, Anna Tambour, of course, Trish Campbell, Medat here. And all these people have been very important and instrumental, as I said, in this conference. We had Ellen Reed, Peter Nickerson, Annette Jackson, Dushka Dragoon, Luis Hidalgo, Dolly Payan, Franz Klaas, and Jean-Luc Topin, who is leading the HA lab in Paris. So I'm very grateful to all these people. I think we achieved a lot of things together. We argued sometimes because we are not perfectly aligned all the time, which is good for science and controversy. But I think that we are making a lot of progress together. So that was Ashi BENF, and I think the BENF schema is now moving and stepping towards molecular technologies for a constant refinement and feedback and enrichment with new members, with biostatisticians, new technologies like natural language processing, AI, patient contextualization, and digital pathology. So this is a very, very exciting development of the BENF conference, and this is a cartoon that was drawn after the BENF actually conference and the publication of the BENF meeting report. 2020 was also the release of the BHOT, which stands for BENF Human Organ Transplant Panel. And we have now these FFPE-based technologies which allow to design this BHOT panel, which covers tissue damage, immune response, and organ rejection genes that we have. And we have this big network now trying to dissect and add that and push that to prove clinical utility. And also we celebrated the 50 years of the human arterial transplantation in 2019. But you know, there is still a long way to go. I think this letter in 2019 really took my attention because someone said that our review article stating that antibodies are a major cause of allograft rejection and long-term allograft failures was not substantiated because the causal relationship between antibody and rejection could not be established. And I took that as a very nice moment to start to think about the evidence and the Bradford-Hill criteria for causation between alloantibodies and long-term allograft deterioration. And I think that this table really prompted us to really understand that we do have a lot of arguments for causality, starting from strength and effect side and reproducibility, but also specificity, a lot of experimental evidence, the temporality, the biological gradient, and the coherence and analogy. So let's not be depressed. I think there is a lot going on in antibody-mediated rejection, and I tend to see more the glass half full rather than the glass half empty. So what's next? What's 2020 and 2030? The future is about integration and contextualization. We are now using well-informed cohort where we have a very holistic approach of past medical history biomarkers. We try to connect patients with their virtual avatar and contextualize that into our reference set. And importantly, this is where the computational resources and modeling, not necessarily AI-based, but also AI-based processing, can lead to build systems that will enhance clinical decision-making process. So that's what we try to build. And you're probably aware of the recent release of the prediction system for risk of allograft loss, the integrated box system, which is summarized here. You know, many collaboration, randomized control trials, many centers, and I'm very grateful to all these authors to try to dissect which were the main determinants and try to build a very universal risk scoring system for long-term prediction of allograft loss. So we are now working hard for implementing iBox in a daily clinical routine practice, from patient application, clinician application, and report generation. And importantly, 2020 has been the step into qualification process through FDA as a surrogate, because we want to use that system also to empower clinical trials and to attract back the industry in our small and tiny field where we can fast track the development of drugs and empower next generation clinical trials. We also want to step into a hard iBox system, and we released this paper recently in Circulation. It's a beautiful collaboration with John Kobachigawa and Jake Patel. And of course, to support that and that huge effort, we are lucky to have two big national and European grants. So this is my transplant journey. It started here, and where I am now, I'm really proud to have this Paul Terasaki prize, where I will be now in 20 years. We don't really know. But what is sure is that we sit on the shoulders of giants, and you recognize all these important persons. We have the way to think that the best way to predict the future was to look back into the past. And I prefer the Abraham Lincoln that the best way to predict your future is to create it. And this is the working family, and let's be stronger together. We are in the Paris group very lucky to work with different people from different fields. We are building important networks, and we try to use this integrative epidemiology to break the barriers between the specialties. Of course, we are relying on a huge network of transplant centers where clinicians are dedicated to patient care, and it's very important. And those integrative strategies is also important for us, and to work, in a very collaborative manner in the Paris transplant group. So speaking of that, I would like to acknowledge the Paris group members from the Kidney Corps, and of course, a special for Carmen Lefaucheur, Olivier Aubert, Christophe Lejean, and Denis Glotz. Jean-Paul Jean, who is a talented pathologist, and we are working closely with him, and many other different people. And this is the Kidney Corps of researchers. And we also have, of course, the lung and heart core with Xavier Jouven, who is also a very instrumental person in the development of the Paris Transplant Group, and many very talented scientists and clinicians and pathologists, as Patrick Grunwald is. And also, we are now expanding, as I said, to the liver, and we are working now with two big centers in Paris. And we also rely on many researchers from different fields, from immunology or data science, mathematics, population science, and it's a very encouraging and to work with different people for different fields. We have a lot of PhD students. It's important to have young blood and to try to make progress with them. These are our PhD students currently doing a PhD in the Paris Transplant Group. We have also master students. And I would like, to make a very special thank you to all our clinical researchers, assistants, and engineers. We have an army of research assistants trying to collect data on a daily basis in every, centers from the Paris group. And of course, we are very grateful to all clinicians taking care of patients. So I would like to thank you for your attention. I would like to thank the Paris Transplant Multi-Organ Transplant Network, because we have many collaboration in France, Europe, North America, and South America. And thank you for your attention.
Thank you, Alex, for, as always, very elegant and thought-provoking talk. And now our next scientific award is the Bernard Amos Distinguished Scientist Award. The Bernard Amos Distinguished Scientist Award was established in 2001 to honor a distinguished Ashi scientist who, like Dr. Bernard Amos, has made significant contributions to our field. The 2020 Bernard Amos Distinguished Scientist Award goes to Dr. Carolyn Hurley. Dr. Hurley is a professor emeritus in the Department of Oncology and Departments of Microbiology and Immunology at Georgetown University in Washington, D.C. Her primary research focus is on HLA and its role in unrelated donor selection for hematopoietic cell transplantation. Her laboratory has reported the nucleotide sequences of many new and confirmatory alleles, investigated HLA population diversity, and explored the functional role of diversity in HLA, T-cell receptor, and natural killer cell immunoglobulin-like receptor, or KIR. On the clinical side, Carolyn oversees a clinical lab performing HLA typing of newly recruited volunteers for the US Unrelated Donor Registry. She served on the Histocompatibility Advisory Group for the National Marrow Donor Program for over 20 years and helped the registry transition its HLA typing from serology to DNA. Dr. Hurley was inducted into the Magis Society as a master teacher at Georgetown University in 2018. She lectured in AFDT or CEOP specialist course for many years, has authored several modules for the Ashi University, and has presented educational lectures at Ashi regional and other national and international meetings. Carolyn has served on the Ashi board and various committees. And I personally witnessed her during the time where we both on the board, and she was instrumental in invigorating the educational committee and many of the educational activities and the Ashi universities in Ashi. Carolyn has served on, she also serves on the WHO HLA Nomenclature Committee and is an international counsel for the International Histocompatibility and Immunogenetics Workshop. She's an active member of the World Marrow Donor Association and has coordinated the development of standards and an accreditation process for the international organization of more than 60 worldwide unrelated hematopoietic cell donor registries. Please welcome Dr. Carolyn Hurley.
For me to present the Bernard Amos Distinguished Scientist lecture. I'd like to thank the awards committee for giving me the opportunity to share my experiences in the HLA field with you and to acknowledge and thank all of you who worked with me on all of these projects I'm going to talk about today. Bernard Amos was an amazing man. He was a role model for basic and translational science. And he made major contributions to the research fields of immunogenetics, tumor immunity, and transplantation immunology. He was able to translate that from basic research into clinical practice. He was the first, one of the co-founders of the first organization in the world to share deceased donor organs based on tissue matching. And he was the president and founder of the International Transplantation Society. Bernard was also concerned about achieving a community consensus in terms of how HLA was tested and how HLA was named. He was the organizer of the first histocompatibility workshop, and he was the first chair of the HLA nomenclature committee. And he was also interested in transmitting that information and all of the information we have on the HLA system. And he was the founder and the first editor-in-chief of our journal, Human Immunology. So like Bernard, I think we all share a set of goals. We have a quest for knowledge, that is that we want to understand the HLA system, the genes that encode HLA, the protein structure, and its function in the immune system. We want to take that knowledge and apply it to the clinic. And then we want to develop best practices in terms of the way that we identify HLA diversity and how we apply it in clinical practice. And finally, we want to share our knowledge with others so that our community can continue to grow and develop, and we want to build a bridge to the future. And I'm going to focus my talk around these four aspects, our shared goals. So we've come a long way in characterizing the HLA system. The HLA system was first discovered in the late 1950s, beginning 1960s, when 3 laboratories discovered that humans, when exposed to foreign tissue, either from transfusion or through pregnancy, would make antibodies that were directed to antigens that were found on the surface of human white blood cells. And it was realized early on that these antigens were polymorphic. And so this led to a joining together of the community to reach a consensus as to how to type these antigens and how to name them. So in those 60 years since that time, we've come a long, long way. And you can see here that now we have the sequence of the entire major histocompatibility complex with all the genes for HLA, plus many more genes that we didn't even know existed at the time. And we've described over almost 28,000 HLA alleles. My story with HLA became as a postdoctoral fellow in the research laboratory of J. Donald Capra. At that time, it wasn't known how many class 2 proteins were expressed by humans. And it was even difficult to convince funding agencies that it was important to fund human studies rather than mouse studies because of their potential application to clinical practice. So my project was to identify the variants or the proteins that were encoded by the class 2 region. We used monoclonal antibodies to isolate HLA proteins. We had monoclonal antibodies that had slightly different specificities for class 2. So we grew transformed cell lines in radioactive amino acids, used the monoclonal antibodies to isolate the class 2 proteins, and then perform protein sequencing in order to characterize the molecules. And this study here that we published in 1982 shows the first 30 amino acids of HLA class 2 molecule, and looking only at 2 amino acids, tyrosine and phenylalanine. And by this data, we were able to show that there were at least 2 different class 2 molecules that were expressed by human cells. We've come a long way since that time, and I'm showing you on the right-hand side of the screen a paper published by my laboratory, first authored by Anna Lazaro, in which we characterized the HLA diversity of individuals that have been typed in our laboratory for the unrelated hematopoietic stem cell donor registry. By that time, we had sequenced almost 100,000 individuals, and we were describing the HLA diversity in that population and many new alleles. And this just shows on the right the sequence of a new allele called A01243. And we were able to determine the complete nucleotide sequence of this allele, both coding and non-coding regions. So things have changed a lot since I was a postdoctoral fellow in terms of our ability to characterize HLA diversity. But sometimes I wonder, maybe we've come too far. Right now, there's about 6,000 or 7,000 alleles at the class 1 loci, and lesser numbers, but still many alleles at the class 2 loci. And I think the reason class 2 is much lower is that these are bigger or longer genes, and so it's much harder to sequence that whole region. So that's really why that class 2 is kind of lagged behind the class 1 in terms of gene numbers. If we take one of the first HLA antigens that was described in the very beginning, MAC, which is HLA-A2, and we look at the diversity that we've described for that particular family of alleles, we can see that, first of all, there's 150 allelic variants that are in the non-coding region. There's 181 variants within the exons, but that do not change the amino acid sequence. There's almost 1,000 alleles in the A2 family itself. We have a number of null alleles, and we also have alleles where we suspect that there's some kind of variation in their expression, but we don't know. Those are the Q alleles. So I think this enormous polymorphism of HLA and our quest to identify those alleles have really led to a lot of problems in HLA typing, and that is that it's very difficult these days to obtain a clear genotype from an individual because of this diversity. This is particularly evident when we're trying to use rapid typing methods to characterize a deceased donor, where the methods you may use are going to result in thousands of alternative genotypes. It's also a problem for hematopoietic stem cell registries because their typings are maintained in a database for probably as much as 40 years. And so obviously the changing understanding of HLA diversity has a big impact on the HLA assignments in registries. So I think it's time for us to turn our attention from stop defining new HLA alleles and spend more time looking at the functional relevance of the alleles we have. I think we want to know how frequent each of the alleles are. We want to know what alleles are functionally relevant that we need to consider in our clinical applications, particularly non-coding variation and variation outside of the antigen recognition domain. We need to understand whether that variation is important to consider. I'm also concerned that we have a lot of alleles that are incompletely sequenced, and then of course those Q alleles where we don't really know their expression. And so I'm concerned that there'll be no continued documentation and repetition of assignment of those alleles and characterization, because functional studies are actually much more difficult than doing allele sequencing. My lab, of course, had been focused on characterizing HLA diversity, so I have to say that I'm one of the contributors to this problem. But in the beginning, when we first started characterizing HLA alleles, we also became very interested in understanding how that allelic variation impacted the function. And I would say that the real insight into that came in a study by Pamela Bjorkman in 1987 when she crystallized the structure of HLA-A2. And I know Pam gave a talk at this meeting probably about this and other structures she's working on. But this really gave us an insight into how the structure of HLA related to its function, and helped bring together studies that showed us that HLA bound peptides, it interacted with T cell receptors, and then eventually that HLA also interacted with other receptors like the natural killer cell receptors we call KIR. My lab was interested in studying that HLA diversity and its impact on function. And one of our first studies, which was shown in the middle panel here, was the difference between DR beta 1 0301 and DR beta 1 0302. We were the first lab to describe that sequence. And Phil Posh, a student in the laboratory, did a number of studies related to understanding how the 4 amino acids that differ between those two alleles impacted peptide binding. And what you see in those graphs in the middle there is basically a series of site-directed mutations of the HLA class 2 protein, and looking between 0301 and 0302, and looking to see how that impacted peptide binding. We were also interested in how T cells recognized HLA, and I was very lucky at that time to be working next door to Sandy Rosen-Bronson, who had generated alloreactive T cell clones. And those T cell clones could distinguish between DRB1 0101 and 0102. Those are two alleles, or two polypeptides that differ by only 2 amino acids. And we wanted to understand the T cell receptor diversity that was being used to recognize what we thought was a very small difference. And so she had a number of alloreactive T cell clones, some of which that saw 0101, some that saw 102, and some that saw both. And we sequenced the T cell receptors from those cells to look at the diversity of T cell receptors. And of course, they were very diverse, which was very surprising because we thought this was a relatively minor difference. Later on, the laboratory delved into the study of KIR, natural killer cell receptors. And on the right-hand side is a study by Will Fraser, where we were looking at the functional differences between different KIR alleles, in this case, 2DL3005 and 2DL3001. And we were looking at how the difference in the sequence between those two receptors impacted their ability to interact with HLA-C. And in the bar graph, you can just see the evaluation of binding using fluorescent beads in terms of binding of the 2DL3 receptors to various HLA-C alleles. And again, we saw that diversity made a difference and impacted, actually not in this experiment, but in others, the affinity of binding between the 2DL3 receptor and HLA-C. I think if I was going on and studied research and doing more research, I would be really interested in the studies we heard about earlier in this meeting about the coevolution of HLA and pathogens or malignant cells. And this is an amazing and very interesting aspect of HLA, how pathogens can alter HLA in a population, and how HLA itself can select for pathogens variation. And I think that's really going to be a very interesting story. I think what's amazing today is we have so many powerful methods to evaluate the HLA system and its impact with the other aspects of the immune response, and just showing you the ability to isolate single cells and study them, more X-ray crystallography, to do various microscopy analyses to see cells interact and exchange materials, to be able to change genes in situ and see their impact on the immune system, and to have access to bioinformatic analysis to allow us to look at very complex pathways of where HLA interacts, in this case in the bottom right corner there, with the various cytokine pathways. So I think this is a very important area that really can use our HLA expertise, and I think we'll have many clinical applications for the HLA laboratory. Coming back to our shared goals, I want to move from basic research to just mentioning the translation of that into clinical applications. And you've heard a lot of talks about that at this meeting, that is applying our knowledge of the HLA system to transplantation and transfusion, to diagnosis of autoimmune diseases, to the determining the risk of adverse drug reactions and immunotherapy. And because a lot has been said about that, I want to move on to talk about best practices. Now, the Ashi has always been concerned about best practices. It has an accreditation program, and many of us are very involved in working within the community to develop a consensus and to develop best practices. And that's what I'd like to focus on next. So we go back to Bernard Amos, and a reminder that he was the organizer of the first International Histocompatibility Workshop and was the first chair of the nomenclature committee. You can see from the nomenclature side, we moved from serologic assignments to DNA-based assignments. And in the first International Histocompatibility Workshop, they tried out a number of methods for identifying HLA antigens. I think we're very lucky that the one of the methods, which was skin grafting, did not win out over complement-mediated microcytotoxicity assays. So I think we're very lucky in that. We got a good start. I became interested in clinical applications in 1992. Up to that point, I had pretty much just focused on research related to the HLA system. In 1992, myself and my colleague Jennifer Ng were invited to an NMDP meeting which was with contract laboratories that were beginning to use DNA typing to type HLA for the volunteer donors going on the United States registry. And this was supported by US Navy funding that was facilitated by Dr. Robert Hartzman. This was a great time. It was early on, and it was a very collaborative group of laboratories sharing reagents and knowledge and expertise. We first got class 2 set up for donor typing, and you can see a paper here with Jennifer as the first author on class 2 typing and showing that it was very accurate and reliable. It took us a longer time to convince people about class 1, making the transition from serology to DNA, because many people thought that serology was great and why change. So we had to actually type over 42,000 individuals in parallel in parallel with serology and DNA-based testing, and looking at the discrepancies that came up and determining which method was at fault. And so the majority of the incorrect assignments were associated with serology, and this was strong evidence that we could go ahead and start routinely typing by DNA in the registry. And Harriet Noreen was the first author on that paper. I'm showing you down here in the corner one of my favorite pictures. We're actually, of course, not really talking about probes and primers. We're drinking margaritas and deciding if we want to smoke some cigars. So it was a wonderful time and a great experience to share with a number of you. That transition from the registry from serologic to DNA typing made a big difference. The accuracy of the typing increased, and you can see how confirmatory typing errors decreased over time in the graph. We were able to increase the number of loci that were included in the testing. We were able to increase the resolution of testing over time. Typing costs decreased, and because we no longer required viable leukocytes, we were able to improve the workflow and the storage of samples, which allowed the registry to type very large numbers of individuals. And so it was a real benefit to make that transition from serology to DNA. Now, one of the important aspects of doing that was that at the time we did that, of course, all the donors previous to that time were typed by serology. So we obviously needed a way to compare DNA assignments to serologic assignments. And so we formed a working group, some of which you can see here in the picture, to make a dictionary to compare serologic types to DNA types. And this was a collaborative effort that was part of the World Health Organization HLA Nomenclature Committee, the UCLA Cell Exchange, the British Rare Cell Exchange, the National Marrow Donor Program, which is the US registry, and expert serologists. I have to credit Martin Myers here, the tall guy in the middle in the back, for developing a neural network which allowed us to predict the serologic types of alleles that had not been characterized by serology. Our first publication of the dictionary came out in 1997. Since then, we published several more editions. We actually quit updating the dictionary because the bone marrow registries no longer needed to do that serologic DNA link because everything was pretty much converted over to DNA typing. But we do know that this dictionary is still in use by the solid organ transplant community and is actually a project in the international workshop to update that dictionary. For those of you who aren't familiar with all the people in the picture, I want to point out our two of our expert serologists that you may not be familiar with. On the left-hand side in the dark black top is Rhonda Holsworth from Australia. And in the middle in front of Martin in kind of the gray and orange top is Eke Schroeder, an expert serologist from the Netherlands. And so we were very lucky to have their help and other help, as well as help from the National Marrow Donor Program and other folks. I think that I was very lucky to become involved with hematopoietic stem cell transplantation and the donor registries, because the unrelated donor registries contain a vast database of global HLA assignments. At the bottom there in the picture of the world, you can see the location of registries around the world, and those million volunteer donor HLA typings. So a huge amount of HLA information coming in from all these registries. And the information that I'm showing you here was compiled by the World Marrow Donor Association, which does a trends report every year on hematopoietic stem cell transplantation and the use of unrelated donors. I want to just give a little shout out to the World Marrow Donor Association, because I think that's been a really key group in terms of achieving consensus among the worldwide registries. This was founded in the late 1980s through 1994 by three well-known individuals. Donald Thomas, who received a Nobel Prize for the clinical application of bone marrow transplantation. John Goldman, who was an expert oncologist who himself performed many transplants, and Jan van Rooyen, our HLA expert, who was one of the first discoverers of the HLA system. And these three men realized that the international collaboration among registries was going to be important in order to find donors, that HLA matching required global collaboration. And they formed this organization in order to develop a standardized practice throughout the world. The WMDA exists today, and most registries are members of it. It holds the database of donor phenotypes, which began as bone marrow donors worldwide under the direction of Jan van Rooyen and other colleagues, but now has transitioned to the WMDA, and it's called their Search and Match Service. And in the bottom graph, you can see that the suggestion by the three founders of WMDA that international collaboration was going to be important has come true. You see in blue and red the contributions, in red of international donors, and in blue the contribution of national donors. And so what we have now is that 50% of the time when a patient gets a transplant from an unrelated donor, that donor comes from a country that's different than the patient. So indeed, the foundation of WMDA and its goals in progress today is aimed at achieving a consensus and standardization throughout registries around the world. One of the things that registries have been able to do is increase the resolution of their HLA typing. New strategies for DNA sequencing have made it possible to type very large numbers of donors. And so the loci typed and the resolution of typing is now at the level that it can be used directly by the physician to select a matched donor for a patient. That is that many of the donors are typed at the level you would like them typed at in order to make a selection of a donor. I'm showing you in these graphs at the bottom some more data from the WMDA that shows the number of loci that have been typed by registries around the world, comparing 2013 with 2019. And I'm using the same color scheme in both graphs. And what you want to see is that in 2013, only 2% of the donors were typed for 6 HLA loci, ABC, DR, DQ, and DP. And in the 6 years time to 2019, that percent has grown from 2% to to 38%. So you can just see the amount of HLA information that's being gathered and utilized by these registries. We took advantage of that, the collaboration through the World Marrow Donor Association and the huge number of individuals that were HLA typed to update the common, intermediate, and well-documented allele list. This shows the 20 registries that contributed their volunteer donor typing. These are random individuals that volunteer to be donors found on the registry, and they are typed by DNA sequencing. We were able to generate get the information for 8 million plus individuals from these donors so that we could analyze the frequency of alleles. And this just shows you one slide from that analysis. This is looking at P group resolution. And so what we can say is that, first of all, for all the known P groups that are listed, or P equivalent groups or P groups that are listed in the IMGT HLA database, 50% of them appeared in the 8 million individuals that were tested. That is among about 15 million alleles that were analyzed. And of that, there was about 25% of the known alleles appeared as common, intermediate, or well-documented. And so you can see that graphed out on this slide here. And so we're hoping that this information, which is now broken down by population group and includes an enormous amount of data, will help refine the HLA typing that goes on from groups around the world. Another important thing that the registries did was to develop very powerful search algorithms. As you know, in the past and still today, HLA experts from laboratories are used to help look at search reports for unrelated hematopoietic stem cell donors and to help predict which incompletely typed donors might possibly turn out as matches for a patient. And here you see two of my mentors, Chinky Johnson and Judy Wade, two serologists that I worked with for quite a while. And they're just an example of the experts in the HLA field that are able to work with transplant centers and give them advice in terms of common alleles and haplotype associations and the likelihood that incompletely typed donors are matched. Unfortunately, not all transplant centers have those experts in place. And so the registries developed search algorithms that could replace those experts. And I'm just showing you here some of the information that's now available on the web to help facilitate searches. So we have from the NMDP, the Be the Match registry, their haplotype and allele frequency data from their studies of their database, and a tool called Haplostats, which is used to predict haplotypes in individuals. In addition, we have those powerful search algorithms that were developed by the NMDP called Haplogic and by the German registry called Optimatch. Optimatch is also used in the search and match registry, the phenotype registry at the WMDA. And these algorithms are incredibly helpful when you have only a few poorly typed potential donors, or when you have many, many poorly typed potential donors. They really can help by predicting which donor you might select for confirmatory typing. I think have really made an incredible difference in our ability to do rapid searches and fairly rapidly identify an appropriate donor for a patient. Finally, in terms of best practices, I want to mention the outcome studies that were done in hematopoietic stem cell transplantation to define who is the best donor for a patient. In 1987, the National Marrow Donor Program established a donor recipient cell repository. And this was under the guidance of Bo DuPont and John Hansen and others who argued that this was going to be incredibly useful in the future. In 1994, there was funding, again, from the US Navy that was provided to retype those donor-recipient pairs using DNA sequencing. And I was lucky enough to be involved in that project as well. And through the support of the CIBMTR, which provided statistical advice, and all of that contract lab sequencing of those donor-recipient pairs, we were able to publish a number of important outcome studies to give guidance to physicians in terms of selection of the best HLA matched donor. I'm showing here the first study by Neil Flamenberg as first author, showing HLA-C was an important allele, a set of alleles to match. And then finally defining an 8 of 8 high resolution match in a paper first by Stephanie Lee et al. In 2007, and then by Joseph Padala, which updated it with a completely separate set of data from the donor-recipient pairs in 2004. And so that provided important guidance to physicians in terms of how to select an HLA-matched donor to achieve optimal outcome. We were also able to use that repository to study other factors that were important in donor selection. And I'm showing you here a publication by Bronwyn Shaw that appeared in 2018, which suggested that using younger donors gave patients a better risk of survival. And so these outcome studies have also been important best practices added into our knowledge of the field. So I come back again to shared goals and just want to touch on sharing knowledge and reminding you again that Bernard Amos organized the first international histocompatibility workshop and was also the founder and editor-in-chief of our journal at Ashi, Human Immunology. I've always been very proud of Ashi because they've really spent a lot of time and energy in terms of educational activities, directed not only to senior researchers and lab directors, but even directed to the newest technologist in your laboratory. And so Ashi has sponsored regional workshops, has an educational platform, is sponsoring journal clubs and so forth, and meetings like this one here. And I think that's been an incredible avenue of knowledge and sharing of information that's been really critical to our field. I want to get close to the end here by talking about how things have changed. When I began working in the field of HLA in the 1970s, Anthony Nolan, shown here in the picture, and his mom, Shirley. Anthony was born with Wiskott-Aldrich syndrome in 1972. He needed a bone marrow transplant, but unfortunately, there was no suitable donor, and he died at the age of 8. Shirley, his mother, wrote a book about her experiences and all the challenges she faced. There were no unrelated donor registries at that time. Class 1 serology was just beginning. HLA-C had just been discovered. Matching was done using the mixed lymphocyte reaction for class 2, and there were no tools and guidance on donor selection. The situation now is very much different. We have over 40 million, almost 40 million volunteer donors, unrelated donors listed in hematopoietic stem cell registries around the world. We also have developed alternative stem cell sources like umbilical cord blood and the use of haploidentical donors. So today we're in a situation where almost essentially every patient has a donor. And this has been facilitated by the collaboration among worldwide registries that has resulted in 50% of donors coming from a different country than the patient. We have very powerful DNA sequencing methods now to characterize HLA class 1 and class 2. We have our outcome studies that can guide us in terms of HLA matching in unrelated donor search, and selection aided by informatics tools and population frequency studies. And for many of these diseases which cause patients to seek a bone marrow transplantation. We have studies that are aimed at understanding what causes the disease. And so for the case of Anthony Nolan with Wiskott-Aldrich syndrome, we know the gene mutation that caused that syndrome. Right now, today, it's treated with hematopoietic stem cell transplantation, and a level of 91% of the patients that receive a transplant survive at 5 years. And gene therapy to correct this mutation is being explored. So we've really come a long, long way in helping patients and their families in the fields that we work in. So going forward, I think there's a few things that I would like to see you do. And the first is to strengthen Ashi's connection with basic science research. When I first started, there was a strong basic research component to Ashi, which kind of was reduced over the following years. And I'd really like to see Ashi turn back more to more basic research, because I think this is where new thoughts and ideas are coming from and new applications that will be moved into clinical application. I would like us to refine our focus on HLA polymorphism so that we can identify relevant variation that impacts clinical outcome. And finally, I would like us to strengthen our worldwide community interactions and perhaps consider standardizing training programs at all levels in order to bridge to the next generation. So my talk was entitled "The Ties That Bind Us Together." And of course, I started out as a molecular biologist here, focusing on molecules and interested in HLA and its role in the immune system. And that's what brings us all together here. But then I talked about shared goals, which we have to increase knowledge of the HLA system, to apply it to the clinic, to develop best practices, and to promote education. And those also bring us all together. But I would say that the ties that really bind us together are our family, our mentors, our colleagues, and our students. And this is just a picture of some of those people that have impacted my life and have helped better the understanding of the HLA system. I'd like to thank you again for giving me this honor, the Bernard Amos Distinguished Scientist Award. I really appreciate it, and it's a great honor to give this lecture in the name of a very remarkable man. And in the end, it's an honor and a joy to be a member of Ashi. Thank you very much.
Thank you very much, Carolyn, for a very moving and hopeful talk. And I particularly appreciate your prescription for what to do moving forward and where we go from here. And I also echo your sentiment about the importance of investing in basic science research. And that's why we created this committee to promote basic science research. And thank you, Carolyn and Alex, and congratulations for winning the award. And if there are any questions from the participants, please, after the session, you can email them to meetings